Hello everyone, welcome back to Biochem with Jayshree. In today's lecture, we are going to learn the TCA cycle. This pathway is also known as the Krebs cycle or the Citric Acid Cycle. The TCA cycle is called the powerhouse of metabolism because it is the major pathway responsible for producing NADH and FADH₂. The TCA cycle is called the powerhouse of metabolism because it is the major pathway responsible for producing NADH and FADH₂, which finally generates ATP through the electron transport chain. In this lecture, we will understand its definition, location, reactions, regulation, energetics, clinical importance, and much more. So let's begin. Before understanding the TCA cycle, let's quickly revise cellular respiration. The process starts with glucose. Glucose undergoes glycolysis in the cytoplasm and forms two molecules of pyruvate. Now these pyruvate molecules enters the mitochondria where they undergo the link reaction, also called oxidative decarboxylation, producing acetyl-CoA. This acetyl-CoA enters the TCA cycle. Inside the TCA cycle, NADH and FADH₂ are produced. Finally, these molecules donate electrons to the electron transport chain where most ATP is synthesized. So remember this simple sequence. First glycolysis, then link reaction, then TCA cycle, then lastly electron transport chain. So this is all about cellular respiration overview. Now move forward. Now understand the definition and different names of the TCA cycle. The TCA cycle is a cyclic series of enzyme catalyzed reactions that completely oxidizes acetyl-CoA into carbon dioxide while producing NADH, FADH₂ and GTP. Now it has three different names. And what are they? First is TCA cycle because the first stable product is citrate, which has 3 carboxylic group in it. That's why it is known as tricarboxylic acid cycle. Second name is citric acid cycle because the first product, which is a stable product formed in this cycle, is citric. That's why the name citric acid cycle. Third name is Krebs cycle. It named after Sir Hans Krebs, who discovered this pathway in 1937. All the three names refer to the same metabolic pathway. Now, the TCA cycle is called the central metabolic pathway because all major nutrients ultimately enter this cycle. And these three major nutrients are carbohydrates, fats, and proteins. When we talk about carbohydrates, it converted into glucose, then pyruvate, and finally form acetyl-CoA. Similarly, fats undergo beta oxidation to directly produce acetyl-CoA. Proteins are broken down into amino acids and many amino acids are converted into TCA intermediates. So, whether we consume carbohydrates, fats or proteins, their metabolism eventually connects with the TCA cycle. That is why this pathway occupies the central position in metabolism. That's why we call it central metabolic pathway. Now comes the location of the TCA cycle. The TCA cycle takes place inside the mitochondrial matrix in eukaryotic cells. Almost all TCA enzymes are located in the matrix. However, there is one important exception, which is succinate dehydrogenase. This enzyme is embedded in the inner mitochondrial membrane. Interestingly, this enzyme also functions as complex 2 of the electron transport chain. Now, what about prokaryotes? Since bacteria do not have mitochondria, the TCA cycle takes place in the cytoplasm. So, in case of eukaryotic cells, it takes place inside the mitochondrial matrix, and in case of prokaryotes, it takes place inside the cytoplasm because they lack mitochondria. Now let's understand the overview of TCA cycle. Acetyl-CoA contains 2 carbon atoms, and when it combines with oxaloacetate, which has 4 carbon atoms, together they form citrate, a 6 carbon compound. Citrate then undergoes a series of 8 enzyme catalyzed reactions. During these reactions, energy is extracted, carbon dioxide is released, and finally oxaloacetate is regenerated. Since oxaloacetate is regenerated, the pathway continues repeatedly, making it a cyclic process. Now that we know the overview of the TCA cycle, let's understand the most important part, the reactions of the cycle. The citric acid cycle consists of 8 enzyme catalyzed reactions. Each reaction has its own enzyme, substrate, product, and significance. Remember one point, the cycle begins when the acetyl-CoA combines with oxaloacetate and it ends when oxaloacetate is regenerated. That is why it is called a cycle. Let's study each reaction one by one. Let's begin with the first step. Step number 1 is formation of citrate. In this step, the reaction is condensation reaction. Here, acetyl-CoA, which contains 2 carbon atoms, combined with oxaloacetate, which has 4 carbon atoms, and together they form citrate, a 6 carbon compound, and the enzyme responsible for this reaction is citrate synthase. During this reaction, coenzyme A is released, and this reaction is irreversible. It is also one of the major regulatory steps of the TCA cycle. This reaction is highly favorable because energy stored in the thiester bond of acetyl-CoA is released. Now step number 2 is formation of isocitrate, and the reaction type is an isomerization reaction. In this step, citrate is converted into isocitrate, and the enzyme responsible for this conversion is aconitase. This conversion occurs in 2 steps. First step is citrate loses one molecule of water to form cis-aconitase, then water is added again but in a different position. As a result, citrate changes into isocitrate. Although both compounds contain 6 carbons, their arrangement changes. This rearrangement prepares the molecule for oxidation in the next step. Now comes the step number 3. Step number 3 is formation of alpha-ketoglutarate, and the reaction type is oxidative decarboxylation reaction, and it is the first oxidative decarboxylation reaction of the TCA cycle. In this reaction, isocitrate is converted into alpha-ketoglutarate, and the enzyme involved in this reaction is isocitrate dehydrogenase. But isocitrate first converted into oxalosuccinate and then alpha-ketoglutarate. In this reaction, isocitrate is oxidized and NAD plus accepts electron and becomes NADH. After oxidation, 1 carbon is removed as carbon dioxide. Therefore, 6 carbon isocitrate becomes 5 carbon alpha-ketoglutarate. And the products removed in this step is 1 NADH and 1 carbon dioxide. This is another important regulatory step of TCA cycle. It is considered the rate limiting enzyme of the citric acid cycle. Now step number 4 is formation of succinyl-CoA, and the reaction type is oxidative decarboxylation. It is the second oxidative decarboxylation. In this step, 5 carbon alpha-ketoglutarate is converted into 4 carbon succinyl-CoA, and the enzyme is alpha-ketoglutarate dehydrogenase complex. This enzyme complex is very similar to the pyruvate dehydrogenase complex. Several cofactors are required for this enzyme. This includes thymine pyrophosphate, lipoic acid, NAD, FAD, NAD+, and coenzyme A. During this reaction, another carbon dioxide molecule is released and NAD+ is reduced to NADH. Finally, a 5 carbon alpha-ketoglutarate becomes 4 carbon succinyl-CoA, and the products are NAD+ and CO2. Step number 5 is formation of succinate. This reaction is called substrate level phosphorylation, and the enzyme responsible for this reaction is succinyl-CoA synthetase. In this reaction, succinyl-CoA converted into succinate. Succinyl-CoA contains a high energy thiester bond. The energy stored in this bond is used to produce GTP. In some tissues, ATP is produced instead. After releasing coenzyme A, succinyl-CoA becomes succinate. And this is the only reaction in the TCA cycle that directly produces ATP equivalent. GTP is the ATP equivalent molecule. Step number 6 is formation of fumarate. Now this is the oxidation reaction in which succinate is oxidized and becomes fumarate, and the enzyme responsible for this reaction is succinate dehydrogenase. This enzyme is unique because it is attached to the inner mitochondrial membrane and it also functions as complex 2 of the electron transport chain. And instead of NAD+, this enzyme uses FAD, and FAD is reduced to FADH₂ and ultimately form a fumarate. So in this step, succinate is oxidized into fumarate and produce one FADH₂ molecule. Now comes the step number 7, which is formation of malate. The reaction type is hydration. In this step, water molecule is added across the double bond of fumarate. As a result, malate is formed. And the enzyme responsible for this hydration reaction is fumarase. In this step, no ATP is produced and no carbon dioxide is released. It is simply the addition of water. Now comes the step number 8, which is regeneration of oxaloacetate. And the reaction type is oxidation. In this step, malate undergoes oxidation and becomes oxaloacetate. Here, NAD plus accepts electrons and becomes NADH, and the enzyme responsible for this reaction is malate dehydrogenase. Now, oxaloacetate is regenerated. This regenerated oxaloacetate again combines with acetyl-CoA and start another round of TCA cycle. And this is the reason why the pathway is called a cycle. Now let's summarize what happens in one complete turn of the TCA cycle. From one acetyl-CoA molecule, the cycle produces 3 NADH, 1 FADH₂, 1 GTP, which is equivalent to ATP, 2 molecules of carbon dioxide, and oxaloacetate is regenerated. Now the cycle is complete. Let's identify the energy producing reactions. NADH is formed in 3 reactions. Step number 3, which is catalyzed by isocitrate dehydrogenase, and step number 4, which is catalyzed by alpha ketoglutarate dehydrogenase, and step number 8 by malate dehydrogenase. GTP is produced only once during step number 5 by succinyl-CoA synthetase, and FADH₂ is produced only once during step 6 by succinate dehydrogenase. Remember this pattern: 3 NADH, 1 FADH₂, and 1 GTP. Now let's understand the energetics of TCA cycle. For every 1 molecule of acetyl-CoA, the TCA cycle produces 3 NADH, 1 FADH₂, and 1 GTP molecule and 2 molecules of carbon dioxide. Since 1 NADH generates about 2.5 ATP, 3 NADH produces 7.5 ATP. 1 FADH₂ gives 1.5 ATP and 1 GTP is equivalent to 1 ATP. So the total energy yield from one turn of the TCA cycle is approximately 10 ATP. So now we can say one turn of TCA cycle oxidizes 1 acetyl-CoA to 2 carbon dioxide, generating 3 NADH, 1 FADH₂, 1 GTP and approximately 10 ATP while regenerating oxaloacetate proteins. Glucose is converted into pyruvate, fatty acids undergo oxidation, and many amino acids are broken down. All of this ultimately form acetyl-CoA, which enters the TCA cycle. Inside the cycle, acetyl-CoA is completely oxidized into carbon dioxide. During this process, high energy electron carriers 3 NADH and 1 FADH₂ are produced along with 1 GTP. These electron carriers enters the electron transport chain where most of the ATP is generated. So in catabolism, The main function of TCA cycle is energy production. Now let's see the anabolic role. The intermediates of the TCA cycle are not only involved in energy production, they also serve as precursor for the synthesis of many important compounds. For example, citrate can move into the cytoplasm and provide acetyl CoA for fatty acid synthesis and cholesterol synthesis. Alpha ketoglutarate is converted into glutamate, which is used to synthesize other amino acids and neurotransmitters. Succinyl CoA is required for the synthesis of heme and essential components of hemoglobin. Oxaloacetate is converted into aspartate, which is used in the synthesis of amino acids, purines, pyrimidines, and nucleotides. Malate can leave the mitochondria and participate in gluconeogenesis, helping the liver produce glucose. So, TCA cycle has a dual role. On one hand, it breaks down acetyl CoA to release energy. On the other hand, it provides intermediates for the synthesis of important biomolecules. That is why the TCA cycle is described as the central amphibolic pathway of metabolism. Now, let's discuss another important concept of the TCA cycle, that is anaplerotic reactions. The word anaplerotic comes from a Greek word meaning to fill up or to replenish. As we learned in the amphibolic nature of the TCA cycle, many intermediates such as oxaloacetate, alpha ketoglutarate, and succinyl CoA are continuously removed from the cycle for the synthesis of amino acids, heme, and other important compounds. If these intermediates are not replaced, the TCA cycle slows down or may even stop because there won't be enough intermediates available to combine with acetyl CoA. Therefore, the body uses anaplerotic reactions to refill these depleted intermediates and keep the cycle running efficiently. If we try to understand the definition, the anaplerotic reactions are metabolic reactions that replenish or restore the intermediates of the TCA cycle, ensuring its continuous operation. Now, let's discuss some major anaplerotic reactions. The most important anaplerotic reaction is the formation of oxaloacetate from pyruvate. The enzyme is pyruvate carboxylase. This reaction requires ATP, carbon dioxide, and biotin as coenzyme. This reaction takes place inside the mitochondria and is activated by acetyl CoA. This is considered the most important anaplerotic reaction because the oxaloacetate is the molecule that combines with acetyl CoA to start the TCA cycle. Second reaction is formation of alpha ketoglutarate. It is the another important anaplerotic reaction. Certain amino acids, especially glutamate, are converted into alpha ketoglutarate by the enzyme glutamate dehydrogenase or through transamination reactions. This helps replenish alpha ketoglutarate whenever it is diverted for amino acid synthesis. Next is formation of succinyl CoA. Succinyl CoA can be replenished from certain amino acids such as valine, isoleucine, methionine, and threonine. These amino acids are converted into propionyl CoA, which is then converted into methylmalonyl CoA, and finally into succinyl CoA. This reaction requires B12 as cofactor. Next is formation of oxaloacetate from the aspartate. The amino acid aspartate can be converted directly into oxaloacetate by a transamination reaction. This provides another pathway for replenishing oxaloacetate. Although the TCA cycle is usually described as closed cycle because oxaloacetate is regenerated at the end of every turn, in living cell it also behaves as an open cycle. But why is it called an open cycle? Because many of the intermediates of the TCA cycle are continuously removed for different biosynthetic processes. At the same time, new intermediates are continuously added back through anaplerotic reactions. For example, citrate leaves the mitochondria for fatty acids and cholesterol synthesis. Alpha ketoglutarate Alpha ketoglutarate is used for the synthesis of glutamate and other amino acids. Succinyl CoA is used for heme synthesis. Oxaloacetate is used for gluconeogenesis and for synthesis of aspartate. Since these intermediates are constantly leaving the cycle, it cannot function as completely closed system. To compensate for this loss, anaplerotic reactions replenish the intermediates. The most important example is the conversion of pyruvate into oxaloacetate by the enzyme pyruvate carboxylase. Therefore, in real cells, the TCA cycle behaves as an open cycle with intermediates continuously entering and leaving according to the metabolic needs of the body. With this, we have completed the citric acid cycle or TCA cycle. Thank you for watching. If you found this video helpful, please like, share and subscribe to Biochem with Jayshree. See you in the next lecture.